In 1838, the United States government forced approximately 16,000 Cherokee people off their ancestral lands in the southeastern United States. They were marched west, through winter, through illness, through starvation. Approximately 4,000 of them died before they reached what the government called Indian Territory in present-day Oklahoma. The event became known as the Trail of Tears. It was one of the most documented forced removals in American history. And yet the people who were removed, the people who died on that march, the people who survived and rebuilt, remained one of the least genetically understood populations on the continent. For 200 years, the story of Cherokee origins followed a simple model. Their ancestors crossed the Bering land bridge from Siberia to Alaska approximately 15,000 years ago. They spread south and east across the continent over thousands of years. They settled the southern Appalachians. They became the Cherokee. That model was the consensus. It was in every textbook. It was taught in every classroom. It was repeated so many times it became invisible, the kind of assumption nobody questions because it seems too obvious to doubt. Then DNA analysis began producing results the model could not explain. And the Cherokee story became one of the most contested genetic puzzles in the history of American archaeology. To understand why the DNA findings matter, you first need to understand what the standard model actually predicts. The Beringian migration model holds that the ancestors of all Native American populations crossed from Northeast Asia into Alaska via the Bering land bridge during the last glacial maximum, approximately 15 to 20,000 years ago, when sea levels were low enough to expose a land corridor between Siberia and Alaska. From there, those founding populations spread south and east across the Americas over thousands of years. The genetic signatures of that migration are well documented. 4 primary mitochondrial DNA haplogroups, A, B, C, and D, are found in Native American populations across the continent and in the Siberian populations that preceded them. These are the expected haplogroups, the ones the model predicts, the ones that confirm the Beringian crossing story. But there's a 5th haplogroup, Haplogroup X, and Haplogroup X does not follow the rules. Mitochondrial DNA haplogroups are inherited exclusively through the maternal line. A mother passes her exact mitochondrial haplogroup to every child, unchanged, across generations. Haplogroup X is found in approximately 3% of all living humans worldwide. In Europe, it is present in populations along the Atlantic coast and in the British Isles. In the Middle East, it is present in Druze, Lebanese, Jordanian, and ancient Levantan populations. In the Caucasus, it is present in Georgian and Armenian populations. In North Africa, small but detectable frequencies. And in North America, concentrated in a specific pattern. The Great Lakes region, the Algonquian-speaking populations of the Northeast, the Ojibwe, the Iroquois, and the Cherokee, the southernmost concentration of Haplogroup X in the entire Americas. Haplogroup X is present in approximately 25% of Cherokee maternal lineages. 1 in 4 Cherokee women carries this haplogroup. The same haplogroup found in the Middle East, Europe, and the Levant. The haplogroup the Beringian model cannot explain. Here is why the presence of haplogroup X in the Cherokee is scientifically extraordinary. The Beringian crossing model requires that any haplogroup found in Native American populations should also be found in the Siberian populations that cross the land bridge. Haplogroups A, B, C, and D are found in both Siberian and Native American populations. The model works for all 4 of them. Haplogroup X does not follow this pattern. Siberian populations show essentially no haplogroup X. The land bridge populations show essentially no. Haplogroup X, the Northeast Asian populations that preceded the crossing, show essentially no haplogroup X. So where did it come from? How did a haplogroup concentrated in the Middle East and Europe end up in 25% of Cherokee maternal lineages? There is no easy answer. There is no consensus answer. What there is a set of competing scientific explanations, each with its own evidence and its own problems. The first explanation is pre-Columbian Atlantic contact. In the 1990s, before genomics had developed sufficiently to resolve the question, researchers at Emory University and the University of Arizona proposed that haplogroup X may have arrived in North America not through Beringia, but through a different route, a transatlantic route. Ancient peoples from the Iberian Peninsula or the British Isles, carrying Haplogroup X, may have crossed the North Atlantic using sea ice edges as a corridor. Similar to the way the Clovis people have been proposed to have crossed, this model, called the Solutrean hypothesis in its archaeological form, suggested cultural and genetic contact between Western Europe and the eastern seaboard of North America, EPTTT 1,000 years ago. If correct, Haplogroup X arrived not from Asia, but from Europe, not through Siberia, but across the Atlantic. The Cherokee, as a southeastern population, might carry the genetic legacy of that ancient Atlantic contact. Most mainstream archaeologists reject the Solutrean hypothesis as insufficiently evidenced. But the genetic presence of Haplogroup X in Cherokee populations is exactly the kind of finding the model would predict. The debate has never been fully closed. The second explanation comes from within the Beringian model itself. Some researchers argue that Haplogroup X was present in Northeast Asian populations 15,000 years ago, but has since been lost from those populations through genetic drift. The random loss of genetic variants in small populations over long periods of time. Under this model, Haplogroup X crossed the Bering land bridge with the original founding populations. It survived in North America through the founder populations that became the Cherokee and Great Lakes tribes. It disappeared from Northeast Asia through random genetic loss over the same 15,000 years. This explanation preserves the standard model without requiring transatlantic contact. It is scientifically plausible. Genetic drift can eliminate haplogroups from populations over thousands of generations. But it requires assuming a very specific and convenient pattern of loss. Haplogroup X surviving everywhere it is unexpected, while disappearing everywhere it would be expected. That convenience makes some researchers uncomfortable. The third explanation is the most recent, and in some ways the most significant. In 2021, a landmark study published in Science sequenced ancient genomes from across the Americas, the most comprehensive ancient DNA analysis of Native American populations ever conducted at that time. The researchers identified what they called a deep population structure within the Americas. Evidence that the founding populations of the Americas were not a single homogeneous group that crossed Beringia and spread uniformly. They were multiple distinct populations with different genetic ancestries, some of which may have taken different routes or arrived at different times. The Cherokee ancestral population, according to this analysis, carried genetic signatures from an ancient founding lineage that was distinct from the populations that produced most other Native American groups. A lineage that partially predates the main Beringian migration, or took a different path through it. A lineage that carried Haplogroup X, not because of European contact or Asian loss, but because the Cherokee descended from a founding population that was always genetically distinct. A population that may have been present in the Americas longer than the standard model accounts for. This finding does not resolve the debate, but it shifts it fundamentally. The question is no longer simply, where did Haplogroup X come from? The question is, who were the people who carried it into the Americas? And how are they related to the people who became the Cherokee? The Y-chromosome evidence adds another layer. Y-chromosome haplogroups inherited through the paternal line tell a different story from the mitochondrial evidence. Cherokee Y-chromosome data shows the expected haplogroups for Native American populations, primarily Q1A, consistent with the Beringian model. But within the Cherokee, and in the work of researcher Donald Yates published through the Journal of Genetic Genealogy, specific Y-chromosome signatures have been identified that carry markers associated with Middle Eastern and Mediterranean populations. Haplogroup T, in particular, a Y-chromosome haplogroup concentrated in the Middle East, the Horn of Africa, and the ancient Levant, has appeared in Cherokee genealogical samples at frequencies that require explanation. Haplogroup T is extraordinarily rare in Native American populations generally. Its presence in Cherokee samples, even at low frequencies, points toward an ancestral connection that the standard model does not account for. The researchers who identified this finding were careful in how they framed it. They were not claiming ancient Middle Eastern people sailed to North Carolina. They were identifying a genetic signal that requires explanation, a signal that combined with the Haplogroup X mitochondrial evidence suggests the Cherokee ancestral population had a genetic heritage more complex than the standard single migration model predicts. The Cherokee oral tradition has always said something different from the textbook model. Cherokee origin stories do not describe a migration across a land bridge from the north. They describe a people who emerged from the land itself, who were always in the southern Appalachians, who had been there since before memory could reach. For generations, scholars dismissed this as mythology. The comfortable assumption was that indigenous origin stories were metaphorical, spiritually meaningful, but historically inaccurate. The DNA evidence is forcing a reconsideration of that assumption. If the Cherokee ancestral population was distinct from the main Beringian founding populations, if their genetic heritage traces to a different founding lineage that either predates the standard migration or arrived through a different route than their oral tradition of deep and ancient connection to that specific land may not be metaphorical at all. It may be genetic memory. The knowledge of a people who understood, through generations of unwritten transmission, that they were different, that their origins were not the same as the people around them, that their connection to the land of the southern Appalachians went deeper than the standard story allowed. The implications of the Cherokee DNA findings extend beyond the Cherokee themselves. If the Beringian model cannot account for Haplogroup X in the Cherokee, it raises questions about what else the model cannot account for. What other genetic signals exist in other Native American populations that have not yet been fully explained? What other founding lineages contributed to the genetic heritage of the Americas that the single migration model misses? Ancient DNA studies of the Americas are still in their early stages compared to the depth of analysis conducted on European and Asian populations. The 2021 science study was a landmark, but it covered a fraction of the ancient genetic variation that exists. Future studies with larger sample sizes, deeper genomic sequencing, and more comprehensive geographic coverage will produce findings that further complicate and enrich the picture. The Cherokee DNA findings are not an anomaly to be explained away. They are a signal pointing toward a more complex and more interesting story of how the Americas were populated. A story that is still being written, still being sequenced, still being debated in laboratories and archaeological sites across the continent. The Cherokee Nation today is the largest federally recognized tribe in the United States. Approximately 450,000 enrolled members, a government, court system, a university, a healthcare system, language preservation program. A people who survived the Trail of Tears, the forced assimilation policies of the 19th and 20th centuries, and generations of systematic erasure. Who rebuilt, who preserved, were still here. Their DNA tells the story of all of that. The survival, the mixing, the adaptation, the continuity. But buried within that genetic record, in the 25% of Cherokee women who carry Haplogroup X, in the Y-chromosome signatures that point toward the Middle East, in the ancient founding lineage identified by the 2021 science study, is an older story. A story about who they were before the Trail of Tears, before European contact, before the written record begins, before the textbook model was invented to explain them. The Cherokee were never who we thought. The DNA proved it, and the full story is still being read.